Uh, Now, today I will give a talk about the complementarity and facilitation in P uptake by intercropping. Actually, this is also my PhD work during when I was in Wageningen in CAU. Um, okay, let's start. First, I want to talk about what is intercropping and why we do intercropping and study intercropping. I think most of you have already be familiar with this. Here I want to give a short introduction. Intercropping is the mixed cultivation of two or more crop species on the same field. And the most common goal of intercropping is to produce a greater yield on a given piece of land by making use of resources or ecological processes that would otherwise not be utilized by a single crop. And there are many different types of intercropping, such as the strip intercropping. That is, two crop species were cultivated in alternative strips, and at least one strip included more than one row. Here we can see in this photo, this is wheat maize, several rows of wheat intercropped with several rows of maize, and also with soybean. And there another intercropping system is a relay intercropping. That is a second crop is planted into an existing crop. For example, with fava bean, which is soon earlier. Fava bean is soon earlier than maize, maize fava bean, and with watermelon intercropping. So wheat is soon earlier than watermelon. And also we can see the right chart. So first the crop is soon first, and later on the second one is coming in. Another intercropping system is the mixed intercropping. I think most of you is common see this intercropping system in Europe. So two or more crop grown together with no distinct row pattern. So two crops sown and harvested together. That is convenient for the machinery. Okay, actually, intercropping has a very long history. For example, in the South and Central of America, especially in Mexico, the meal pie is very famous. It contains maize, bean, and pumpkin in one system, and it is called as Three Sisters. Mm. And also in China, intercropping has a history of more than 2,000 years. And it is the earliest documentation is about in many very old books like 齐民要术 and 农政全书. 齐民要术 this book is the earliest agricultural we can say it's the earliest agricultural encyclopedia in China. This book documented many different cropping systems and agricultural technology in Wei dynasty. So it's about thousands years ago. In this book, it systematically summarized the advanced experience of intercropping in this book. So intercropping has a lot of intercropping advantages. It produces higher resource capture and productivity, build up of soil organic matter, and pest and disease suppression, and some can also reduce the climatic risk. In this photo, it's a very beautiful landscape in Gansu province in China, and we can see this is the Yellow River in the middle. On the other side, there is no crop, so a lot of sands. It's yellow, and on this side, it's a lot of green with intercropping and crop tree mixture. So it can produce a lot of not only green, but also food for people living in this area. Actually, I work in a very big group working on intercropping. Actually, this work is started by Phosphorus Zhang in 1989 when he got his PhD from Germany in the University of Hohenheim. His thesis is about understanding the role of sulfurs to mobilize iron in the soil. So after he back to China, he wrote some books on the methodologies on the soil and the plant nutrition, and he wants to relate his expertise with some interesting stories in the field. Because they found that in the farmers' field, for example, in the central part of China, the soil is calcareous, and the peanut is facing some iron deficiency. We can see the monoculture peanut with some yellow leaves. But interesting phenomenon is that the peanut leaves are green when they grow neighboring with maize. So it's a very interesting phenomenon. So he supervised with a PhD student, Yuan Mei, to do this work, to use his knowledge on the sulfur to mobilize iron by the maize to the peanut. And then in 1990, around 4, so Professor Li Long worked in Gansu province. In this province, intercropping is also very popular, and the farmers grow a lot of intercropping like wheat maize and maize fiber bean. It shows a very high yield production. So based on these phenomena in the field, we also want to understand why. So Professor Li Long did his PhD thesis with Phosphorus Zhang, and to understand the interspecific facilitation and the competitive interactions between the intercropped species. So this is the start of intercropping study in CAU, and I also did my PhD and also my master on studies on intercropping in CAU. So across the last more than 30 years, we did a lot of work in the lead of Phosphorus and also Professor Li Long, and get a lot of deeper understanding of on why there is overyielding in nutrient uptake and production. And we also did a lot of work to do modeling work in collaboration with Wageningen, and also do some quantitative analysis like meta-analysis working with Wodka, and some long-term experiments that were led by Professor Li Long and got a lot of achievements. Okay. So for my talk, actually today is about phosphorus. So I want to go to 3 parts. So first, the advantages of phosphorus uptake by intercropping, and then for the mechanisms on facilitation, the complementarity in uptake by intercropping. In the end, is our perspectives and conclusions. For phosphorus, I think most of you are familiar. It's an element very important for plant growth, but the phosphorus use efficiency is very low because even farmers apply a lot of phosphorus, but most of them are fixed by the soil. So the phosphorus use efficiency is very low. In China, it's around 25 to 30%. And we can see from this map, a lot of phosphorus are surplus in the soil, especially in China. So that's why we want to study phosphorus uptake by intercropping. So we want to know how to increase this P uptake by crops. whether we can do that by intercropping. So previous studies showed that actually this is one meta-analysis. It shows that intercropping can increase the P uptake. The average net effect of P uptake of cereal legume intercropping is 3.67 kilogram P per hectare. The net effect is calculated with the difference of P uptake, the observed P uptake and the expected P uptake. Here, the observed total P uptake was calculated as the sum of the species P uptake, while the expected P uptake was calculated as the sum of the species P uptake multiplied by their land share in intercropping. Is this clear for you? Yeah. Okay. So the results means that intercropping can take up more P uptake than they're expected from the monocultures. So that means there is P overyielding by intercropping. Okay, that is the outcome. So what about the mechanisms? Still with a meta-analysis work done by I and Wolfgang, and we showed that the complementarity effect largely contributed to the overyielding in intercropping. So here, this complementarity effect is also calculated with some formula. Actually, that is quantification on the outcome, but not on the mechanisms. For the complementarity effect, it consists a lot of complementarity courses, like the niche differentiation, niche partitioning. So that means different species use different resources or at different time or spatial. And it's such as the resource or spatial temporal complementarity or facilitation. I will go to in detail. So for these two aspects. So in this talk, I will go to in detail about the resource complementarity and the facilitation. So what are the definitions of complementarity and facilitation in resource use? In this figure, in the left, we can see if two crop species use the single source, there will be competition, right? But if two crop species use different resources, because in the soil or for the light, they can use different resources at different time, so there will be complementarity. And for the facilitation, that means there are different resources, and one species can use them in different. The other one can use that resource very easily and help the other one to also use the unavailable resources. For example, for the phosphorus, there are soluble phosphorus, so both species can use. But for the immobilized phosphorus, the cereal cannot use them effectively, but the legume can use them very efficiently, so they can mobilize phosphorus to the other species. So what about intercropping? And previous study showed that, and they also found that in the yield, the overyielding, there is overyielding, and the overyielding was mostly caused by the below ground interactions. Because this study was done by Professor Li and published in 1999. And actually, this is the first study to show intercropping overyielding is caused by the below ground interactions. In this field experiment, they did 3 treatments. So they separated the root of fiber bean and the neighboring maize with different sheets. So the first one is the plastic sheet. so no interaction between two roots. And the nylon net, so it's a mesh. So the roots cannot transfer, and there is no root interactions, but like the chemical root exudates can transfer. The other one is no separation, so there is root interactions. So let's see the results. So for the system, for the intercropping without a root interaction, the land equivalent ratio is around 1. But with a mesh, so with the chemical transfer interaction of the exchange of root exudates, there is overyielding. The LER is larger than 1. And without separation, the land equivalent ratio is larger. So based on these results, we think the below ground interaction play a key role in for the overyielding. Then we want to ask why the below ground interaction can contribute to the overyielding. For their follow-up studies, they determined the root exudates of maize and faba bean, and they found that the faba bean roots can acidify their rhizosphere soil, and we can see from this photo. So the different colors represent the pH of their rhizosphere soil. The yellow means there is a strong acidification. The purple means no acidification. Then we found that the faba bean root can acidify the rhizosphere soil strongly, because the acidification then can release the fixed phosphorus by the soil phase. So that means the faba bean can mobilize the sparingly soluble soil, sparingly soluble phosphorus in the soil, and then facilitate the P uptake by the maize. This is for the inorganic phosphorus. Then they also did other work about whether can intercropping facilitate the P uptake from the organic phosphorus. Because for the organic phosphorus, plant can only use it by exuding the phosphatase, either acid phosphatase, then it can hydrolyze the organic phosphorus to the available P pole. So then plant roots can use that. And still in this kind of design, they use plastic sheet separation, the mesh separation, and no separation. They found that the maize chickpea intercropping. So when there is no root competition, but only the exchange of root exudates, the total P uptake was highest. So and they determined the acid phosphatase activity, and they found that the chickpea acid phosphatase activity is much higher than maize. So that means the chickpea can mobilize the organic phosphorus in the soil, and the neighboring maize can benefit from that. So this is the mechanisms on facilitation, and we can see clearly from this figure. So that means the faba bean release some root exudates like the phytase or phosphatase to mobilize organic phosphorus, or to release some proteins to acidify the soil, then to mobilize the calcium P, or the faba bean can release the carboxylase to mobilize also the calcium P or the iron P and the aluminum P. Then all the mobilized P go to the available people. So these two crops can share the people, then to increase the P uptake. So this is the mechanism on phosphorus. And they also found that when the fiber bean can mobilize the phosphorus, so there is phosphorus patches in the soil, and the neighboring maize, which has a very strong root growth ability, then they can grow a lot of roots to take up the phosphorus from the mobilized pea caused by their neighboring fiber bean. And in this study, actually, there are a lot of data. The main idea is to show that the maize root length is much higher than the fiber bean. So that means the maize can take up the phosphorus easily, and the fiber bean can play a role to mobilize the phosphorus. Okay. So in addition to the facilitation, what about the complementarity? Actually, these two mechanisms are very difficult to differentiate, but we tried to focus on the complementarity in one study. So the complementarity in nitrogen uptake is already well known. I think it's clear for you. So the cereal can take up the soil organic nitrogen. The legume can use the nitrogen in the atmosphere by nitrogen fixation. And a lot of studies have shown this fact. Then we wonder, can we also test the complementarity in pea uptake by intercropping? Because we know there are different pea pools in the soil. So we hypothesized that whether two intercropped species can use different pea pools, like the organic pea or the inorganic pea. So we did this pot experiments during my PhD study. We want to test, first we want to test the ability of crop species to take up pea from different pea sources. So we selected 13 crop species that are commonly used in the intercropping system, such as the wheat, maize, millet, rice, and white lupin, fiber bean, chickpea, peanut, soybean, and also other crops like cabbage and potato. Then we grew them in monoculture and supplied them without pea or with the most available pea or with the sparingly soluble pea like calcium pea, iron pea, and phytate pea. So sorry for the display of the figure because of the connection. The cable.
You missing something?
Yes, you see here like this, but
there's big ovals here that you cannot see.
yeah. Okay. No problem. I can explain.
You can with your hand.
Oh, okay. So yeah. So we compare the each species, whether how they can use the calcium pea, phytate pea, and iron pea. Actually, we calculated a ratio. So the ratio is calculated as the pea uptake from each sparingly soluble pea divided by the most available pea. If the ratio is around 1, that means this species can use calcium pea, phytate pea, or iron pea very effectively as the most available pea. That means its pea uptake ability from the sparingly pea sources is very high. So here we found two species, millet and chickpea. So millet can use calcium pea effectively, but not for the chickpea. The chickpea can use more phytate than the calcium. Then we want to combine these two species together and see whether the mixture can take up more pea. So we designed these treatments, so 9 treatments. So we grow plant species in monoculture or in the mixture and supplied with single pea and mixed pea. And we suppose that if the species mixture growing on the mixed P sources take up more P than the mixture growing on the single P. That means there is overyielding due to the complementarity in using different P pools. Okay, let's see the results. Is there complementarity? These two bars are the expected values. So they expected millet and the chickpea growing on calcium, single calcium P and phytate P. And we also did control treatments that with maize. They are they show very similar P uptake abilities on different P sources. Then we suppose the wheat maize will decrease the P uptake. And we found that for wheat maize, yes, that the results verified our hypothesis. So millet chickpea mixture growing on the mixed P sources took up more P than, sorry, yes?
Okay, quick question. What, perhaps I'm not sure if it's a super question, but what exactly is phytate phosphorus? Is it organic?
Yeah, organic.
Okay. Right.
But for millet, we also found that even they have similar P uptake abilities, they still show overyielding in P uptake. But here we found that is not because of the complementarity in tapping to P pools, but because wheat is more competitive than maize. So wheat took up more P, but that is more than the loss of maize. So there is still overyielding. Then we did several experiments in pots and also in the field to test whether our selected species combinations indeed show P uptake overyielding. So in the pot experiments and also in the field experiments, we do monocultures and intercropping under low P apply and under the mixed P apply. And we, well, okay, let's see the results. Is there complementarity in P uptake and overyielding? First, we determined the rhizosphere processes, because the P uptake abilities is based on their abilities to release the root exudates. And we found that for chickpea, yes, it released a lot of carboxylates, but also acid phosphatase activity, because for the legume, if they can use the phosphorus, they needed to release carboxylates. But for the organic phosphorus, they also needed to release carboxylates to replace the organic phosphorus fixed by the soil phase. So that means chickpea can mobilize the phosphorus because its ability to release carboxylates is higher than the millet. So we suppose there were facilitation in P uptake. But in the field experiment, we only found this P overyielding in one season, in the second season, but not in the first season. But in the first season, the P uptake overyielding did not lead to the overyielding in the green. So that means the P uptake overyielding did not transfer to the P uptake to the green overyielding. So that means the complementarity and facilitation with respect to P uptake was not the only driver to the overyielding in the green yield in this relay strip intercropping. So is there any other explanation to the P uptake overyielding under the low P conditions? Because we know that the facilitation by the root via the root exudates relies on the root overlapping. But in the field, there is a strip intercropping actually only for the border row intercropped species, the root overlapping is a lot. But for the inner row crops, the root overlapping is less. But we know that there is mycorrhizal fungi, the hyphae about several hundred meters per gram of soil. So we were thinking how the mycorrhizal networks contributed to improve the P uptake by intercropping. So we did this pot experiment in the end. So in this pot experiment, we have the treatments like monoculture and the mixture without mycorrhizal fungi and with mycorrhizal fungi. And we also did a mesh for the root compartments. And with the root mesh, also there is no root direct interaction, but the fungus, the hyphae can transfer, and the root exudates also can transfer in between the two root compartments. And also for the intercropped species, intercropped treatments, so we only inoculate one species in one side, because if we can determine the root colonization by the fungi in the other side, that means there is a common mycorrhizal network is formed. Okay, so these are the experiments when we harvested. So it's clear that without AMF, the plant growth is not so good. But with AMF, either with one side or the other side, the plant growth is better. And we found that without AMF, there is no overyielding in the P uptake. And with AMF, only with the chickpeas inoculated, there is P uptake, but not for when the millet is inoculated. And in this situation, when the chickpea is inoculated with mycorrhizal fungi, chickpea is the donor of this mycorrhizal network, and the millet is the receiver of the mycorrhizal network. And in this condition, both species can benefit in the P uptake. So why was that? Then we determined the NP ratio, and we found that without AMF, the NP ratio is higher than 20. That means the plant is under P limitation. So the chickpea is under P limitation. But with AMF, the P limitation is released because the mycorrhizal fungi can transfer a lot of phosphorus to the above ground. But without P limitation, there is nitrogen limitation for the legume, for the chickpea. Then this nitrogen limitation stimulates the legume to fix more nitrogen from the atmosphere. So there is also nitrogen uptake, also complementarity and nitrogen use in this system. So that's also contributed to so in this situation, so more root exudates from the legume could be, because we found that when the chickpea is the donor, because here the chickpea has larger biomass than the millet. So in this situation, it fixes more nitrogen, and more root exudates are released in the rhizosphere, and the root exudates can act as a carbon source for the microbes. And here we determine the more phosphorus solubilizing bacteria in the rhizosphere. So we think that not only the mycorrhizal fungi, but also some bacteria played a role. And in the follow-up, one of my master students did another pot experiment, also with a mesh, But here she also found that only with AMF there is P uptake overyielding, and she found that there are also some rhizosphere bacteria in this when there is when the plant is inoculated with mycorrhizal fungi, especially some phylum are related to the phosphate solubilizing bacteria, like the bacillus. So we think there is not only root exudates, not only mycorrhizal fungi, but also some bacteria. Okay, in sum up of the above content, so we proposed this opinion. We think that for the intercropping, when compared with the monoculture, there is cascading biotic interactions below ground. So the below ground interaction is very complicated, and we want to decipher the interaction. And proposed this systematic perspective. And here we think that compared with the monoculture, we know that the above ground photosynthesis at the carbon will go beyond one part of the carbon will for the root growth, and the other part of the carbon will go for the rhizosphere, so the root exudates. And the other carbon will go to the mycorrhizal fungi, because the mycorrhizal fungi is rely on the carbon from the plants. Then all these three pathway can mobilize or take up the phosphorus and nutrients to the above ground. So this is closed loop for plants. So this is for the monoculture. What about for the intercropping? So what about when we include one legume in the monoculture system? And we think that so with the addition of a legume, because a legume can release a lot of carboxylates, phosphatase, so it can release some phosphorus. So this is a legume roots. It can release some phosphorus. And will form a P patches between two roots. And the maize, the cereals can grow the roots very fast and a lot of thin roots. Then it can take up the phosphorus and more nutrients above ground, and then more carbon, relatively more carbon will go below ground, and some more carbon will support the root exudates further of the maize, and also more carbon go below ground, and some root exudates act as the carbon source for the microbe. So this is a kind of cascading effect. And can we use this? So yes.
In your previous slide, the figure that mainly focuses on the benefit of maize from the legume, but does the legume also benefit from maize growing legume?
Yes, actually, legume also benefit from maize to fix the nitrogen by the nodulation. But here I did not go to in details. Yeah, legume also benefit. Okay, we want to expand, use this perspective to explain what we see in the field. Because in the field, this is a very typical photo for the intercropping. So here is wheat intercropped neighboring with maize. But maize is depressed by the wheat, because wheat is soon earlier and very plant height and no light, less light for the maize. But also because the below ground, the wheat root system is very large. It's also very competitive with the maize. But let's see, when maize neighboring with the fava bean, the maize grows better than the inner row maize. So we think maybe some thing is going underground. So what's that? So in summary of our previous studies and what I mentioned before, so we can understand like this. So this is a maize growing alone. So the blue lines represent how the maize can use the phosphorus in the soil. So by growing roots or release also some exudates, even less than the legume. This is the circle of how the maize take up the nutrients. But for the intercropping, when the fiber bean is coming in, so the fiber bean can release root exudates to mobilize either the mineral bound phosphorus, the organic phosphorus, and also the organic phosphorus by the phosphatases, also to the available people. Then the neighboring maize can use root proliferation, where a lot of thin roots to take up the available people. Then more root exudates will go down, and the root exudates, the root exudation is increased either by releasing the phosphatase to use organic P, or can recruit some microbes like the phosphatate solubilizing bacteria to also mobilize the organic P and also the mineral bound P, then to increase the available P pool. So this is also like a loop. Okay, in summary, so facilitation and complementarity in P uptake actually underpin the P overyielding by intercropping, and the mycorrhizal networks strengthen the facilitation and the complementarity in P uptake by intercropping. And the multi-interface interactions encompassing the root-to-root, root-rhizosphere, and the rhizosphere microbiome interfaces drive the cascading effects and contributing to the increased productivity in intercropping. Okay, finally, I want to thank my supervisors during my PhD and also my collaborators and my co-authors and also my students.